my dear Bacha Party. I hope that you are doing wonderful, you're happy, you're healthy, and everything is nice. Well, I welcome you all to PU2 Brahmastra series, but today we are going to deal with the chapter that is microbes in human welfare. And let me tell you, I have a heartfelt gratitude to all of you who attended the previous session. I felt so good. I felt so good being there in the live session, answering to your things, chatting with you in the live chat. Now, that is something that I want. That vigor I want from you guys, that participation I want from you guys, that makes me so happy. So do not forget to be like that in this live session also. Ask me doubt, tell me your answers. I'm so happy when you tell the answers. Whether it be wrong or right, it does not matter. Your efforts matter to me. I always tell that. So keep on doing that. And also, if you love the video, do not forget to show your love in the comment section also. Would love to see you, see the comments, see you also basically later on when I go through the sessions. I feel so happy. So that's all. Let me stop my buck and let me go ahead. Why did I choose this chapter? Because microbes in human welfare hold 7 marks, my dear children, and according to the 3 blueprints that we have. Okay, the 7 marks has been divided separately. For example, according to blueprint 1, there will be 2 1-marker question, 1 2-marker question, and 1 3-marker question. According to blueprint 2, there is 2 1-marker question and 1 5-marker question. According to blueprint 3, there is 2 1-marker question, 1 2-marker question, 1 3-marker question. So you can basically expect any type of questions coming off from this particular chapter. If it's a 5-marker, there will be only 1 5-marker, no 2-marker, okay? Because it's of 7 marks. So anyways, there should be only 1 5-marker question. Fine. So there is one very important announcement. We have launched the PU2 Success Blueprint Test series. Okay. So today it will be 14th. Tomorrow on 15th you would get your first question paper. Okay, and the syllabus we have already shown up, okay, in the YouTube channel itself. Now, if you have registered, you have enrolled by paying this much amount, that is 599 rupees, you might have got it in your WhatsApp community channel also, the schedule for this. So, according to the schedule, you can prepare and then give the test that would be released tomorrow. So, over here in this particular series, we are going to give you the video solutions and the doubt clarification classes as well. So, do not forget to enroll. If you are interested, the link is given in the description box. Do not forget to go ahead with it. Now, the number of questions I have included is 11 of 1-marker question, 2 2-marker question, 4 3-marker question, and 3 5-marker question. So, I've included all the questions, the repeated type of questions that have come so far, and also the questions from model question paper. So, if you are someone who tried to solve the model question paper, okay, and you are finding difficulty, then you would find it really easy because I would be explaining each and everything in much detail. Okay, and one more disclaimer, there are so many names of bacteria, fungi and stuff like that, scientific names that you might not be able to recollect very easily. So, I might be repeating them often so that you at least remember it by their names. And here there is one exclusive thing. So until now I was never writing the MCQ answers. Okay, like I was telling the MCQ answers and I will showing you, but over here I have pre-written the MCQ answers as well as a little bit of explanation. So if you are going through the PDF, you will get an explanation to it as well. Okay, now the first question says, which of the following water samples in the table given below will have higher concentration of organic matter? Which of them, these water samples, will have higher concentration of what? Organic matter. Now children, if there is too much of organic matter, you need too much of what? You need too much of bacteria as well. Okay, what bacteria? Basically you need some aerobic bacteria probably. Anyways, you need some bacteria to break it down. And these bacteria, these aerobic bacteria are going to consume a lot of oxygen. Aerobic as in it needs oxygen, right? So it is going to consume a lot of oxygen. Okay? If there is too much amount of organic matter, there will be too many amount of aerobic bacteria and there will be a requirement of too much of oxygen as well. And this requirement is known as BOD, biochemical oxygen demand. So, biochemical oxygen demand, if it is more, that means the organisms which are breaking down, they need more oxygen. Cool? So, basically, if I have to create a situation like this, that means the BOD level would be really high. And also, if BOD level is high, if organic matter is high, what I am trying to say, don't you think the pollution that is there, it is also high? The pollution will also be high. So what I have to search? I have to search for the value of BOD. I have to search if it is high or not. So basically, if it is high, I say this is the correct answer. So here I see there are two options with high BOD. That means there is high amount of oxygen required. Okay. Now the level of pollution should also be high, right? The level of pollution should also be high. Then only this thing can be attained. Okay. So level of organic matter, if it is high, that means the level of population is high. Not population, pollution is high. So here I see high is there. Here I see high is there. But here BOD is low, so I cannot say this is the answer. Here I see both of them are high and this becomes the answer. Okay. So in a water sample where the level or the basically concentration of organic matter is high, that means you're talking about pollution. Pollution is also high. Okay, it is highly polluted. That is why we say organic matter is high. Now to break down this organic matter, you need lots and lots of aerobic bacteria, and this aerobic bacteria will be needing lots and lots of oxygen, which is why we say the biochemical oxygen demand would be really, really high. So this becomes your answer to the question. I hope I was able to explain you this. If I was able to explain you this, give me a thumbs up. Okay, if you have a doubt, tell me in the comment section. Cool. So your answers goes like this. So this is the explanation that I was talking about, that I have given explanation for each and everything. So BOD is what? A measure of amount of oxygen required by microorganisms to break down the organic matter in water. So how much organic matter is there? That much amount of oxygen they require, that is known as BOD. High BOD means high biological oxygen demand, means they need, that means that particular area has high amount of organic matter, and for that they need high amount of bacteria which need high amount of oxygen as well. Okay, so that indicates high level of organic matter which requires more oxygen to decompose and leads to water pollution. It will lead to water pollution. Now one more very important thing, when there is high oxygen demand by aerobic bacteria, okay, they are taking in all the oxygen for breaking down things. What happens? All the other chunumunu organisms which are there in the water, they become deficient of oxygen. They're like, give me some oxygen, you're taking everything. So basically you're depleting oxygen and you are harming the aquatic life which is normally over there. Which is why you say polluted water, no organism can stay, because there is high BOD. Now low BOD means the water is cleaner and requires lesser oxygen for microbial breakdown, indicating better water quality. So this would be the answer for also a question which state define BOD. This is a normal question that have come so many times, okay, which is why I have included the answer over here itself. Cool. So this becomes your answer. I hope you have understood the concept. Let's go on to the next question. An agricultural labor was spraying. An agricultural labor was spraying some powder mixed with water, okay, some powder mixed with water onto what? Onto fruit trees to get rid of insect larvae. He wanted to kill insect larvae. Which of the following biocontrol agent could have been used here? Biocontrol agent, that means you are not taking the help of pesticides and things like that to kill the pest. Instead, you are taking organisms. These organisms are going to eat the pest. So this is a biocontrol agent. Okay, this is a biocontrol agent. Now, your sole motive is to kill what? The insect larvae. So you have to choose one among them which is going to affect the insect larvae. And the answer, there is a straight answer over here. The answer is Bacillus thuringiensis, which is also known as BT. So bacteria, that is Bacillus thuringiensis, is a bacteria, okay? What it does is that it releases and it has inactivated toxin in it. It has inactive toxin in it. Okay, what happens when the insects, they come and feed on a plant or something, they are going to take in this bacteria also. If the bacteria is also there, it is going to be taken up. Now the bacteria comes inside the insect's gut. This is the gut of the insect, suppose. This is the insect. Okay, now it comes in the gut. What happens? This particular bacteria's inactive toxin which is there, it becomes active, because what happens? The gut of the insect, it is having alkaline pH, a basic pH, and basic pH is that pH when the inactive toxin becomes active, and now it is going to affect the gut of the organism and later kills it. So this is how Bacillus thuringiensis works, okay? And when we are putting this gene which is going to create some toxin and stuff like that, we are putting this gene from the Bacillus thuringiensis into plants, so the plants can also produce the same toxin. It is known as Bt cotton or Bt brinjal or things like that. Okay, so Bacillus thuringiensis is the answer. Trichoderma is not going to kill the insect larvae. Trichoderma is a fungus which is going to kill other fungus, which are parasites. Okay, Trichoderma is a fungus which is going to kill other fungus. Now over here you have dragonfly. Dragonfly basically is there It is there to kill the larvae of mosquito. Even though mosquito is an insect, but over here they have set fruit trees to get rid of insect larvae. It comes mostly, the answer becomes this only. This is more accurate answer. Dragonfly feeds on specific thing, that is mosquito larvae. Now ladybird is something that feeds on something that is known as aphids. Okay, if you do not know aphids, you should be googling it right now. See the picture, you will remember it a lot better. Okay, ladybird has a beetle, say red and black beetle that is there, which goes and eats aphids, and aphids are something that feeds on plant and stuff like that. So this becomes your answer. All clear? With this, can you answer other questions also? Okay, if this and all comes. Okay, dragonfly comes and eat mosquitoes larvae. That's all. Now, so biocontrol agent is what? A living organism used to control the population of harmful pests, diseases or weeds in an ecosystem. Cool? Now, if a patient has undergone myocardial infarction, it is actually infarction, okay, anyways, leading to heart attack. To remove clots from the blood vessels of the patient, which enzyme is used by the doctor to treat the patient? So basically, when someone is having heart attack, that means the muscles of their body, the muscles of the heart muscles are not receiving enough amount of oxygen, probably due to some clots being made up over here, which is why the blood is not getting transported, and if blood is not getting transported, it's not going to transport the oxygen also, because blood cells carries the oxygen, right? Now if I want to dilute this clot, if I want to remove this clot, what should they do? What enzyme is used by the doctor to treat the patient? Pectinase, it is used to break down pectin. Okay, protease, which is used to break down protein, lipase, which is used to break down lipids. But over here you have streptokinase. This becomes the most specific answer. What is it, children? Streptokinase. This is something that will break down the clot. Okay, streptokinase has an indirect effect. It is going to convert plasminogen. This thing you do not have to learn, okay? Plasminogen into plasmin. Plasmin, my name is Jasmine, this is plasmin, okay. And this plasmin is going to basically break down another thing that is known as fibrin, which is a major part of the clot, So basically streptokinase is going to break down the clots anyhow. That is something that you have to remember. Now streptokinase you get from a bacteria that is known as Streptococcus. Streptokinase, where do you get it from? Streptococcus. Will you forget this? Streptokinase from Streptococcus. I will be asking you after 5 more questions. Cool. So streptokinase is produced by the bacterium Streptococcus and are modified by genetic engineering, is used as a clot buster. It is known as clot buster. Please remember this term. Clot buster, okay, for removing clots from the blood vessels of the patients who have undergone myocardial infarction leading to heart attack. Cool. Next question. Which one of the following alcoholic drinks is produced without distillation? What do you mean by distillation? So you should be knowing all these key terms, otherwise you're basically mugging up things, and I don't want you to mug up things. So you have to find the alcoholic drink which is produced without distillation. Distillation means basically, suppose you have some drink, okay, you are going to make it heated and things like that, and this particular drink is going to vaporize. Okay, suppose in this particular jar there is alcohol and water also and something, okay. So distillation basically means I am going to treat it in a way such that the alcohol which is there, it is going to vaporize, and these vapors are collected in a tube, and these vapors later will be giving rise to a fluid over here. So over here, if you see the fluid which is made over here, it has more amount of alcohol. It would be having more amount of alcohol because this is just a vapor form that has come over here and has been like this. It has totally vaporized. The water is over here. Okay, so here you would be having a lot of other things. You will be having water vapor also. You would be having a lot of other things, but more amount of alcohol will be there. It is more volatile. You get it? So here, in these type of drinks which have underwent distillation, the amount of alcohol would be comparatively more. So you are basically asked which alcoholic drinks are produced without distillation. That means here the amount of alcohol content is what? Would be comparatively lesser. If it has underwent distillation, it will be more. So wine, do you think it has more amount of alcohol content? So understand the basics, you will understand everything. You don't have to remember the things. So wine is something that everyone drinks. Do you think it has lots of alcohol in it? No. But whiskey, rum, brandy and all, it has lots and lots of alcohol content in it. Okay, now there is one more thing. Apart from wine, there is also one more guy. There is also one more guy which has less, that is beer. that is beer. So wine and beer is given in your NCERT. So wine and beer are produced without distillation, whereas whiskey, brandy and rum are produced by distillation of the fermented broth. Okay, the fermentation is done and then this thing has been taken up. Next, bioactive molecules, that is cyclosporin A, is produced by. Cyclosporin A is produced by a guy you studied earlier also. Can you guess? Over here, there is the guy's name. Over here, the guy's name is there. Can you guess who is that guy? Okay, cyclosporin A is a thing that is being produced by a guy over here. Any guesses? Well, its name is Trichoderma polysporum. Okay, its name is Trichoderma polysporum. Cool. And it is basically, I told you, it's a fungus which basically kills other fungus. So this is a fungus. Bioactive molecule cyclosporin A is produced by a fungus, that is Trichoderma polysporum, which has a function. It is an immunosuppressive agent, my dear children. So whenever you have organ transplant and stuff like that, what happens is that our body thinks it is some foreign particle because I am getting it transplanted from some other organism, right? From other organism basically means a fellow human being. But what happens, anything that comes as a foreign particle, my body will be like, I have to fight with it. Even though it is useful for us, but our body thinks that it's a foreign thing, we have to anyway fight with it. So at this point of time, we have to make sure that our immune system, okay, has to be a bit suppressed so that that guy can take some time to adjust in our body, okay, and then be a part of us. So cyclosporin A is used as an immunosuppressive agent in organ transplantation, and it is produced by the fungus Trichoderma polysporum. Okay, Trichoderma polysporum. So you study about trichoderma in two places. One is basically in biocontrol agent, and the second is over here when you're talking about cyclosporin A. It is immunosuppressive agent, cyclosporin. Cool? Chalo. Next, Swiss cheese with large hole is produced from. Cheesy. It has large hole. Where do you think it is produced from? Okay, I'll tell you something. Okay, this bacteria has a lot of serum in it. Okay, it is too shy. It is known as Propionibacterium shermani. Propionibacterium shermani. So I remember it like that. Okay, so it is too shy. So Swiss cheese is produced from the bacterium that is Propionibacterium shermani. It's a bacteria, so you just add prop. So how do you write it? Propioni. Okay, Propioni bacterium. Okay, it's a bacterium, and then you write the Shermani part. It is Shermani, which has two I, my dear children. Okay, I am writing it like this so that you also write it. Have your pen and paper, write the names which I am going to tell you now. It will be helpful for you. It will be definitely helpful for you. Just write it anywhere, at the back of the book or anywhere. Okay, just writing practice. So Propioni Shermani, it is very properly doing Shermana. Okay, whatever you want to say. So why does it have the spaces over there? Because this guy is going to basically produce a lot of carbon dioxide. When it is going to react with it, it's going to produce a lot of carbon dioxide, and when this carbon dioxide goes out, basically it is going to create pores, which is why it has large holes. So the answer for this is what? Propioni shermani. Propionibacterium shermani. Don't forget the bacterium part. An example for mycorrhiza forming fungi is, what do you mean by mycorrhiza? Mycorrhiza is basically a symbiotic association. Is a symbiotic association. Symbiotic association basically means you're kind of helpful towards each other. So symbiotic association is happening between the plants and the fungus. This symbiotic association has a specific name. It is known as myco. It has double R, one H and one C. Mycorrhiza. So an example of mycorrhiza forming fungi is glomus. Is what? Glomus. From the species that is glomus. So it looks like glomus, okay. So what happens over here, the fungus which is there, no, it will basically help in, okay, fungus, you know, it has lots and lots of hyphae. It has lots and lots of hyphae. And this hyphae, no, it can penetrate inside the soil and things like that, and it can easily exchange what gases. So basically it takes in gases that is phosphorus and stuff like that, and then say that my dearest friend plant, you can have it. You can have it. So plant is getting phosphorus and everything with the help of this association. Also, you know, examples like trichoderma, which is a fungus, what was it doing, children? Trichoderma was basically, it's a fungus and it kills other fungus which is going to affect the roots of the plant. So the roots of the plant are also here. the roots of the plant are also here and they are in kind of in association, right? So basically what happens, this guy, the fungus which is there is going to feed on those funguses also and going to kill them. So basically in any ways, if we see, the plants are kind of protected with the help of them. And the plants, what do they do? Plants also provide certain nutrients and stuff like that. Okay, a place to stay. Okay, a place to stay and stuff like that. Works? So this is mycorrhiza, children, and the answer is glomus. I have written over here, let's see. So fungi are also known as to form symbiotic association with plants. This is known mycorrhiza. Many members of the genus, okay, many members of the genus glomus from mycorrhiza, the fungal symbiont in this associations absorb phosphorus from the soil and pass to the plant. Plants having such associations show other benefits also, such as resistance to root-borne pathogens. I told you, right, in the roots there will be pathogens. So these plants will be showing resistance to that. They will be able to survive, okay. Tolerance to salinity and drought as well. So these are the things that the fungi partner provides it for them. Cool. So it is going to forming genus is from the genus glomus. Okay, cool. Chalo, next. Dash is used for fermenting malted cereals and fruit juices to produce ethanol. Okay, fermentation means basically what? In anaerobic condition, you are going to break down organic matter, basically some sugar, organic matter to alcohol, to CO2 and some amount of energy. This is known as fermentation. And all of this thing has to happen in what? Basically an area where there is lack of oxygen and aerobic condition should be maintained over here. So dash is used for fermenting malted cereals and fruit juices to produce ethanol. What is being used for fermenting this? There should be something to be used. Okay, for this fermentation to happen, okay, there are some microorganisms needed, or else this fermentation won't happen. This breakdown of things won't be happening. So what are those organisms? So the answer for this is your yeast, that is Saccharomyces cerevisiae. So spelling is this. If I am wrong by any chance, I don't think I am wrong. Okay, so Saccharomyces cerevisiae is the answer. Saccharomyces cerevisiae is the common yeast that you have. Okay, what do you mean by malted cereals, my dear children? Cereals means any pulses that you have. Okay, so malted cereals means usually they are in dry state, but what are we going to do? We are going to help in germination. We are going to provide little amount of water. They will slowly start germinating. What happens when these pulses start germinating? All the enzymes within them becomes active. All the enzymes which is there within them, they become active, and these enzymes are required for breaking down the organic matter. So basically we are going to help in germination, and then later we are going to dry it. So all the enzymes are now active, and now we are going to use those enzymes for breaking down of what? Organic matter. But over here you need Saccharomyces cerevisiae. Do not forget that. That is one most important thing. Okay. In yeast only fermentation will happen. So some yeast, Saccharomyces cerevisiae, used for bread making and commonly called as brewer's yeast, is used for fermenting malted cereals and fruit juices to produce ethanol. What do you call this? Brewer's yeast. Okay, it is used in bread making and all, and also for fermentation. Lots and lots of work. Cool? Chalo, next question. Write the scientific name of the fungus which produces cyclosporin A. Tell me. I told you I will be asking you after some time. Tell me this. Write the scientific name of the fungus which produces cyclosporin A. It has divert function. What was the name of the fungus, my dear children? Trichoderma polysporum. Always while writing, please remember that the P should be, in the second term, the P should be, specific name should be small over here. T should be in capitals, this should be in small letters. Done? Chalo. Now, which bacteria is commonly found in anaerobic sludge during sewage treatment? Which bacteria is found in anaerobic sludge during the sewage treatment? Whenever you are dealing with the dirty things and basically you want to clear it up, there's an anaerobic sludge being found. Okay, a sludge where basically there is lack of oxygen. Okay, you have a lot of things, you have to still purify it, but you are doing it in an area where there is lack of oxygen. And when there is lack of oxygen, there is a group of bacteria which becomes really important. This is methanobacterium. It is what? Methanobacterium. Basically, you call them methanogens. You call them methanogens, but the name of the bacterium is methanobacterium. Okay, spelling is like this. Methanobacterium. Methanobacterium. What do they do, children? Methanobacterium is going to take the organic matter and break it down into certain gaseous things such as CH4, which is known as what? Methane and things like that, which is why they are going to release gases, and these gases could be used. So while we are treating the sewage, we are giving back the purified water and stuff like that. We are going also producing a lot amount of biogas. So these CH4 methane which is there, it can be used as a biogas, and this biogas can be used in the kitchens, it can be used for electricity purposes and a lot of other things. So basically in anaerobic condition, this particular bacteria is really active and really efficient, where it is going to break down the organic matter, whatever is there. Okay, the organic matter, whatever is there, into gaseous format, that is CH4 and stuff like that. So Methanobacterium is commonly found in anaerobic sludge during sewage treatment. These bacteria are also present in the rumen, which is a part of the stomach in the case of cattle. So cattle have a part of the stomach which is known as rumen. They are also found there. So I am associating the functions and making you remember things so that you do not forget. So do not think like, okay, Methanobacteria I studied there, how can it be here? So it becomes easy for you to run. You know Trichoderma's two functions, right? It fights with the root fungus, and second thing, it produces cyclosporin A, which is acting as an immunosuppressive agent during organ transplant. Over here, Methanobacterium which is there, okay, it is going to work in anaerobic condition. Okay, they are the ones that can work in any bad conditions. You can remember like that. So Methanobacterium are going to work in anaerobic condition, and they are going to produce gaseous things like such as CH4, which is methane, which is why their name is Methanobacterium. And this CH4 basically can be used as biogas. Now this same Methanobacterium is found in the cattle also. Ma'am, why there? Basically in the cattle they feed on plants. Agreed? They feed on plants, my dear children, and plants basically, their cells are made up of cell wall. The cell wall is made up of something that is known as cellulose. It is known as cellulose, and this cellulose which is there, okay, this cellulose which is there has to be broken down in the case of cattle, and who does it? Methanobacterium does it. Okay, so a lot of cellulosic material present in the food to cattle is also present in the rumen. In the rumen, these bacteria help in the breakdown of the cellulose. Works? Okay. Chalo. Children, we are going to enter the 2-marker question. Before that, I am going to ask you a question. Are you able to understand things? Am I a bit faster, or is it okay? Okay, if you're understanding things, do show your love to me. I would love to see that in the comment section. Yes. Now, what are biofertilizers? Also, if you have a doubt, ask me. Okay. What are biofertilizers? Mention its significance. Tell me. What do you think? What do you think are biofertilizers? So, what is the use of fertilizers? Fertilizers core use is to provide nourishment. Okay. Fertilizers have a rich content of NPK, that is nitrogen, phosphorus and potassium. So I want the plant to have all this thing. Now when I say biofertilizers, probably I am going to talk about organisms which are going to do this job for me. They are going to probably fix the nitrogen, the phosphorus and things like that for the plants. So the plants which is there, they can grow very easily, and you have to mention their significance. So you can go there and answer like this. Biofertilizers are organisms that enrich the nutrient quality of the soil. The main sources of biofertilizers are bacteria, the fungi and the cyanobacteria. Example is rhizobium. The most common thing that you can find. Rhizobium, the plant cannot take the atmospheric nitrogen. No, dear children, they cannot take the atmospheric nitrogen because it is in this format. It has triple bond and the plant cannot break it down. So basically there are bacteria in the soil which would fix this by converting it to something else, probably converting into ammonia, and then ammonia can be treated to nitrates or nitrites, and then probably this can be used by the plants. So this function is being done by bacteria like this. Okay, what do you have? Basically for availability of nitrogen you have rhizobium and azotobacter. Remember the names, azotobacter, and for phosphorus we have other things. So basically biofertilizers are nothing. They are the ones that enrich the nutrient quality of the soil. Okay, they are the ones that fix nitrogen, they fix phosphorus, they fix potassium and stuff like that. Okay, and who are those people? Who are those organisms? It can be a bacteria, it can be a fungi, it can be a cyanobacteria as well, which is bacteria itself. Now, nutrient availability, it is of significance, it is increasing the nutrient availability. So, increase the availability of nitrogen done by rhizobium, and the other example is what? Azotobacter, and also phosphorus and also potassium. Okay, done? This is a 2-marker question. Write in your points. This should be point 1, this should be point 2. First explain biofertilizers, then say what do they do. They are going to increase the nutrient availability. Give the examples. Without examples, nothing is possible. Underline the examples as well. Azotobacter, rhizopia. Now, define BOD. We have learned about BOD previously. What is BOD, children? Anyone in the comment section, tell me. BOD stands for biochemical oxygen demand. A question to you. If a water body is polluted, does it have high BOD or does it have low BOD? Does it have high BOD or low BOD? It will have high BOD. Why? This is polluted. If it is polluted, that means more organic matter. More organic matter, then to break it down, you need more aerobic bacteria. If you need more aerobic bacteria, these aerobic bacteria will be needing more oxygen. And if they are needing more oxygen, can I just say that the level of BOD is high? Because BOD means what? Biochemical oxygen demand. So their demand for oxygen is really high. So we can suggest, or we can just write, okay, before that, telling you the definition before doing that, mention the commercial importance of Monascus purpureus. Do you know the commercial importance of Monascus purpureus? Purpureus, basically, it is something that is going to give you statins. So Monascus purpureus, no, it gives you statins, and statins, you can remember, statin stabilizes your cholesterol. Stabilizes. Oh my god, I am not able to remember the spelling of stabilizes. Okay, which is actually a sad situation, but okay. It makes the cholesterol level stable. You can remember like that, cholesterol level stable. Okay, you can remember like that. So basically, how does it do that? There is an enzyme which is there required for the formation of cholesterol. Okay, there's an enzyme which is required for the formation of cholesterol. It kind of basically inhibit the formation of that. It is responsible for inhibiting the enzyme responsible for synthesis of the cholesterol. Okay, it inhibits the enzyme which is responsible for the production of what? Cholesterol. Cholesterol is an organic compound which is there in our body. Okay, I hope you know that. And stabilizes, the spelling is like this. Okay, lysis. Yes, this is spelling. Cool? Chalo. So that is a function of Monascus purpureus. Now we will read BOD and Monascus purpureus together. So BOD refers to the amount of oxygen. First you have to write like BOD refers to complete the statement. Biochemical oxygen demand. Biochemical oxygen demand. Then you say the rest, that it refers to the amount of oxygen required to oxidize or basically break down the total organic matter by the bacteria. Amount of oxygen required by the bacteria to break down the organic matter. Either way, in what? In 1 liter of water. In 1 liter of water, very much important. In 1 liter of water. So high BOD means high oxygen demand. High oxygen demand means there are so many bacteria. That means there is so much organic compound. That means there is pollution. Now, what does Monascus purpureus do? It produces statins which act as blood cholesterol lowering agent. It lowers blood cholesterol level by competitively inhibiting. It competitively inhibits the enzyme responsible for synthesis of cholesterol. So it says, before you come, I am going to do something about it. I am not allowing the synthesis of cholesterol. So this is how it works. Okay, works. Monascus purpureus. Okay, chalo. Next question. This is a 3-marker question, my dear children. Complete the below given tabular column with appropriate answers. Dash is there and it produces lactic acid. And what is the use of lactic acid? Who is there? Lactic acid is being produced by a bacteria which is known as lactobacillus. they come under lactic acid bacteria category. So there are a group of bacteria which produces this. So they come under this category, that is lactic acid bacteria. Lactobacillus is an example. It produces lactic acid. Okay, and what does this lactic acid do, my dear children? It helps in forming curd. It helps in forming curd. How does it do that? Basically, in the milk. Okay, in the milk, there is something that is known as lactose, which is the sugar present. This get converted into lactic acid with this bacteria only. And then this guy, this lactic acid, when this lactic acid is there in the milk, this is the sugar which is there. In the milk, there is also a protein which is known as casein. It is known as casein. So what happens in the presence of lactic acid? That means the acid level is really high. The level of acid is really high. In this particular level, you are basically disturbing the pH. When you say it is too acidic, what is the pH, children? pH should be low. Too acidic means pH is low. Too basic means pH is high. So in this particular low pH, what happens? The structure of the protein is going to change. The structure of casein is going to change, which is why it forms small lumps like this, which is known as the curd lumps, which is produced. So lactobacillus is there for basically producing lactic acid, which is going to form the curd. Now methanogens. Methanogens, what are they going to produce? Methanogens, methanobacterium. Remember, what is the product there? Methane. The gas methane. And what is it used? It is used as what? Biogas. Cool. Works. Remember. Methanogens. Example, methanobacterium. Now next, EF, treatment of bacterial disease. So we just know it is there for treatment of bacterial disease. So treatment of bacterial disease, that means you're talking about an antibiotic. So can I just name an antibiotic here? Becomes a lot more easy. So have you heard of penicillin? Have you heard about penicillin? So penicillin is an antibiotic, okay, which is taken out from a fungus whose name is Penicillium notatum. Okay, whose name is Penicillium notatum. So we'll write penicillin. Just 2L over here. Penicillin. Okay, and there is Penicillium notatum. Okay, spelling I also forget sometimes. Okay, Penicillium notatum. No tantrum. Penicillium, no tantrum. You can remember like that. But don't write no tantrum and there and come, please. Okay, it's going to be your exam. So penicillin is released by Penicillium notatum, and that is used for treatment of bacterial diseases, my dear children. So your answer goes like this properly if you write. So lactobacillus is there for lactic acid production, which coagulate and partially digest the milk proteins. Next thing, you have methanogens, which produces methane. It is used as biogas. Penicillin, which is there, it is produced by Penicillium notatum, and it is used for treatment of bacterial disease. Now I'll tell you a fun fact. This is an antibiotic. Not everyone is okay with penicillin. For example, there are people who are allergic to penicillin. Okay, so I had some some issue. Okay, so I had everyone basically have antibiotics in their life, right? So I also had, and doctor asked me, Jasmine, do you have allergy to penicillin? I said, no, I don't have allergy. How would I know? Basically I said no. Okay, I got chill. What will happen? Basically I was small also. So and then what happened? Not that small anyways. So I was given penicillin, and later on I was not able to breathe properly. So people have allergy to penicillin, okay, and all the other antibiotics as well. But yes, it's just a fun way to remember things. So remember, Jasmine ma'am had allergy to this. So penicillin is there, which is released from Penicillium notatum, a fungus. Cool. Will you forget this? And then Monascus purpureus. What was it releasing? Statins. Statins stabilizes, okay, the blood cholesterol level. It actually lowers down, but okay. Explain the role of microbes in industrial products. In industrial products, there are so many microbes name. So over here you go, the name. So production of microbial products useful to human beings on an industrial scale requires growing microbes in a very large vessel called fermenters. Okay, and these are the names of the bacteria. Sorry, of the organism. Aspergillus niger, it releases citric acid. Aspergillus niger gives out citric acid. Acetobacterium aceti, acetic acid. Clostridium butylicum, butyric acid. Lactobacillus you studied. So whatever we studied, we are going to mark it in red. Lactobacillus, lactic acid. Saccharomyces cerevisiae, it is there for ethanol and also for bread formation. Penicillium notatum we just studied. Okay. Streptococcus, streptokinase is being produced. What does it do? What does it do, children? Remember the function also, please. Clot buster. Okay, this is an antibiotic. This guy is also known as brewer's yeast. If you remember like this, then only you will be studying. Okay. Now Trichoderma polysporum also you studied. Listen, it becomes so easy for you now, no? Cyclosporin 8 is being produced, and Monascus purpureus tetans. Now if you would have seen it like this only, you would have gone mad. Now you have seen it in parts, it becomes a lot more easy. We just have to learn these three names. Will we be able to know? Will we be able to learn it? Aspergillus niger, citric acid. Acetobacterium aceti, acetic acid. Clostridium butylicum, butyric acid. Okay, Aspergillus niger, citric acid, Acetobacter aceti, acetic, Acetobacter aceti, acetic acid. Same name, no? Clostridium butylicum, butylicum is there, so butyric acid. Aspergillus niger, citric acid. That is something different, but okay. I'll ask you after 3 minutes. Okay? Mark the countdown. Now, explain the role of microbes in household food products. So, what are those microbes which are there in household? So, these are industrial stuff. Okay, these are industrial stuff. I want industries to produce all this stuff, right? Now, if you're talking about food products, household food products, what are those? Lactic acid bacteria, they are there. Okay, and the other thing that you study, Saccharomyces cerevisiae is there. Apart from that, do you know anything else? Let's see. So over here we have lactic acid bacteria which grows in milk and convert it into curd. Lactic acid bacteria produce acid that coagulate and partially digest milk proteins. What are those milk proteins? We also know that, ma'am, that is casein. And what is this acid being produced? Lactic acid. And where is this being produced from? From lactose. A small amount of curd added to fresh milk as inoculums or starter. Matlab, if you have the curd, okay, the curd has the bacterium, no? So if you're putting the curd in the milk, then again this particular bacteria will feed on the milk protein and things like that. Not the milk protein, milk sugar, and then feed on that and grow there. So that is why we put inoculums. Inoculums as in the starter thing that are being put. Now LAB, that is lactic acid bacteria, improves nutritional quality of the milk by increasing vitamin B12. Okay, now baker's yeast is there, that is Saccharomyces cerevisiae. It is also known as brewer's yeast. Brewer's yeast, baker's yeast, everything is there. So baker's yeast, Saccharomyces cerevisiae, is used for making bread. Okay, then toddy, which is there. It's a traditional drink. It is made by fermentation of sap from the palms. Sap from the palms. So what is it? Okay, anyways. Large holes in the Swiss cheese are due to production of what? CO2. Who does it? Propionine. Oh, how did I forget it? Propionine shermani. Propionine bacterium shermani, okay, which produces what? Large amount of CO2, which is why it has large holes. It is found in Swiss cheese. So it becomes a major big player in the formation of Swiss cheese as well. Now there is one more cheese which is known as Roquefort cheese, kind of very costly. Okay, now this is ripened by specific fungi. I searched for the fungi's name. It is Penicillium roqueforti. Roquefort is a name, Roqueforti is the name of the fungi. Okay, Penicillium roqueforti. Okay, it gives specific flavor to it. So how many names do you have? Lactic acid bacteria, okay, which is going to basically help in the curdling of the milk, and then baker's yeast or brewer's yeast, that is your Saccharomyces cerevisiae, which is used to make the bread. The toddy is being made from the sap of the palm, okay, and then you have Swiss cheese, the holes over there, done by the production of carbon dioxide, which is produced by what? The guy that is Propionine bacterium shermani, okay, and then you have this guy, oh, his Roquefort cheese, okay, which is kind of costly cheese. This is from the ripening of it has been done with the help of a fungus, which is the fungus. Remember, it's a fungus, okay. It is known as Penicillium roqueforti. Cool. Rock shorty, you can remember like that. Anyways, so that is there. Okay, I hope I am helping you in some way. I can teach you in the organic way, but in that way it becomes, that is why I am energetic today, because I don't want to teach you in organic way. You won't be able to remember it. I am pretty sure of that. So, and any questions can arise from any part of this things that I am telling you right now. So which is why I am stressing and repeating so that you remember it. Okay, chalo, next. 3-marker question. Explain the role of 3 organisms as biocontrol agents. Now biocontrol agents as in organisms which are going to control the pest, So 3 organisms name I have to say. Okay, you have Bacillus thuringiensis. First name, Bacillus thuringiensis. BT, the bacterium, the bacteria which is there. Okay, going to kill the insect larvae and everything. Other organisms name you want? What name do you have? Dragonfly. Dragonfly was basically killing other flying organism, which is mosquito. But what about mosquito? Basically the larvae of the mosquito. Okay, the larvae of the mosquito they're going to act like a pest, right, for us. So yes, that is there. Now apart from that, ladybird, the cute little ladybird which is there, which is not a bird but a beetle. Okay, it is going to feed on aphids. I hope you have searched by now. It is going to feed on aphids. They asked for 3. Can I say one more? Children, my cute little champions, tell me. Trichoderma, the fungus, it is going to act on the fungal pests. Where? In the root of the plants. They are all going to act like biocontrol agents. I don't know why this question is there. Please remove it out. Okay. Now, what is the significance of BOD? This question has repeated so many times. I told you. Significance of BOD is what? Basically, you would be able to analyze the level of pollution. The level of pollution, biochemical oxygen demand, tells you how much polluted it is. If it is way too much, BOD is way too much, that means it is has quite a lot of organic matter and hence it is way too much polluted. Okay, and if it is less, that means it is less pollution. Now give an example for a fungus found in mycorrhiza. This was a 1 marker question coming from the fill in the blanks. Remember, mycorrhiza is what? Association of plant and the fungus. Okay, fungus is providing what? The phosphorus and everything. What is the plant doing? It is providing nourishment and stuff like that. But basically, tell me the name from the genus Glomus. Cool. Chalo, read it. So, the use of biocontrol measures will greatly reduce our dependence on toxic chemicals. Yes, that is why we are using. Because if you are using pesticides and all, do you remember there was a pesticide that is DDT. Okay, DDT, which was found in Kerala. Okay, and what happened in Kerala? In Kerala, there is a district that is known as Kasaragod. And any Malayalis up here? Okay, anyways, I am a Malayali. Okay, so in that particular area, what happened? People were using a lot of pesticides, that is DDT, and later on this DDT seeped into the soil and things like that. And obviously it goes into water and people drink water, and lots and lots of people were affected because of this. They had mental retardation problem. Lots of people lost their life because of this thing, and then it was totally banned. Okay, so there are so many problems associated with pesticides that we are using because they are chemical products. Now, if you have biocontrol agents, basically we are stopping our reliance or dependence on the toxic chemical substances, which is kind of really good thing, right? So, ladybird is there, a familiar beetle with red and black marking, and dragonflies are there used to get rid of aphids and mosquitoes. So, aphids is ladybird and mosquito larvae is basically the dragonfly. Then we have Bacillus thuringiensis. It is used to control butterfly, caterpillars or any insect larvae. Okay, dried spores are mixed with water and sprayed onto vulnerable plants. Okay, the spores of the particular bacteria are being mixed with water and then sprayed, okay, on plants such as brassicas and fruit trees, where these are eaten by the insect larvae. In the gut of the larvae, the toxin is released and the larvae gets killed. I explained you. Trichoderma, a free-living fungus used to control several plant pathogens is there. Okay, basically what is it controlling? The fungus only, other type of fungus which is going to affect. Now, baculovirus is there. That is something that you have not studied. Okay, baculoviruses are pathogen that attack insects and other arthropods. Arthropods basically means, arthropods is a bigger unit. Under arthropods comes insects. Okay, apart from insects, there will be lots of arachnids like spiders and stuff like that, all of them there. Okay, so basically your spiders are not insects. Please remember that. Insect is a different category under arthropods. So, baculoviruses are basically those that attacks insects as well as arthropods. The majority of baculoviruses used as biocontrol agents are in the genus Nucleopolyhedrovirus. Please remember. So, this baculoviruses, okay, the majority of them are from this particular genus, Nucleopolyhedro. Yeah, Nucleopolyhedrovirus. Okay, Nucleopolyhedrovirus. Okay, so here we have baculoviruses as well. They asked for 3, we have given for more so that you remember. Now, these viruses are excellent candidates for species-specific narrow spectrum insecticidal application. They have no negative impact on plant, mammals, birds, fish, etc. This is very useful in integrating pest management. Okay, so they are kind of species-specific. So, if you want something perfect, you can go for baculoviruses. Now, BOD is a measure of organic matter present in the water and also an indicator of the pollution. I told you significance. Give an example of fungus found in mycorrhiza. You have the fungus from the genus Glomus. This is the genus. Okay, what is the genus of baculovirus which is most famous? Nucleopolyhedro. Nucleopolyhedro viruses. Okay. Now we are up with the 5-marker question, my little champions. Let's see. With respect to the microbial products, its source and uses, identify A, B and C in the following table. Define BOD. Again BOD. We are not going to define BOD. What is BOD? Tell me. Tell me in the session. What did you understand by BOD? And one question to you. Which is that particular organism that gives you citric acid? Which is that particular organism which gives you citric acid? We studied it. And I told I will be asking you within 3 minutes. So I might have moved away from the time zone, but yes. So children, you answer me that, okay, and also use your brain over here. Cyclosporin A, where do you get it from? Cyclosporin A, where do you get it from? It is acting as an immunosuppressant. You get it from Trichoderma polysporum, which is a fungus. Monascus purpureus is something that controls the blood cholesterol. It is acting as a blood cholesterol lowering agent. What is this thing? What does it produce? Statins for stabilizing. It competitively inhibits the enzyme which is there for the making of the cholesterol pigment. Not pigment, cholesterol unit. Now streptokinase is there, which is produced by Streptococcus. What does it act as? Streptokinase, it is going to break down something, right? It is going to act like clot busters. Okay. Now define BOD, biochemical oxygen demand. I hope you have written the answer. I'll check in the comment section. Okay. Now BOD, biochemical oxygen demands, means the same thing, that is the amount of oxygen required by the aerobic bacteria for breaking down of organic matter in 1 liter of water. That is BOD. Now what do you mean by flux? What do you mean by flux? So during sewage treatment, during sewage treatment, I'll tell you a situation. So basically there comes a situation where there is lots and lots of waste water and stuff like that, and you are going to treat it in aerobic conditions. So there are aerobic bacteria over there. Okay, there are aerobic bacteria over there. So while you're treating, you will be finding certain clumps over here, aggregates over here. Okay, and these things are made with the help of bacteria as well as some fungal filaments, These things that are created is known as flux, my dear children. It is known as the flux. So over here, flux are clumps or clusters of fungal filaments, bacterial cells and other things also that are held together by extracellular substances produced by the microorganisms. So these guys only are releasing certain things which makes them stay together. These aggregates are involved in biodegradation of organic matter during processes like wastewater treatment. So these are kind of useful. Okay, they would be there helping in the biodegradation of the organic matter which is there in this particular thing. In aerobic condition, they are going to help us out. Bacteria plus fungal filaments, the clusters they are forming, it is known as what? Flux. Okay, so you can write your answer like this and please check your answer. Cyclosporin A, Trichoderma polysporum is there. It is acting as an immunosuppressant. Statin is there, Monascus purpureus and blood cholesterol lowering agent. Then you have Streptokinase, Streptococcus, clot buster. We have studied this. BOD is what? The amount of oxygen required by microorganism to decompose the organic matter present in a given water sample. Given water sample ni 1 liter of water. Okay, and flux are this that we just studied. Now citric acid is produced by Aspergillus niger. Aspergillus niger. Okay, if you were not able to answer the question, which is why I have told you. Now 14th question, explain a typical biogas plant with a neat labeled diagram. What do you mean by biogas plant? So biogas plant is where you are going to produce the biogas. Okay, and if you want to explain it with the help of diagram, it looks like this. We'll understand it first and then only we'll go ahead. So first thing first, just give me a second, okay? Okay, anyways, so over here you have a mixing tank. you have a mixing tank. Okay, I was looking for the terms because I forgot the terms. Okay, in this mixing tank, you are going to put the dung and whatever bio waste is there in your household, whatever waste are being made. Okay, for example, you have the chica of any plant or organism, not organism, basically any fruit or anything like that, any covering of those. Okay, or not the plastic covers, I'm talking about the covering of the fruits and stuff like that, and also the cow dung and everything. So we are going to make a slurry out of it. By what? By mixing the dung with the water. Okay, now there is a pipe which is known as inlet pipe. You have to use your imagination over here. Okay, inlet pipe. Cool. This is there. Through the inlet pipe, things are sent to the digester. This is where the organic matter is getting broken down. This is where the organic matter will be broken down. And who does it? There are so many. This situation over here, it is maintained, it is made sure. Okay, over here, you are making sure that basically this cow dung which you are putting, this dung that you are putting, it has high amount of methanogens already in them. They have a high amount of methanogens already in them. Now they got a perfect situation over here. That is anaerobic situation. That means you are going to make sure that the oxygen is not being transported over here. This is a closed situation. So this is what, children, anaerobic situation that you are creating over here. Anaerobic situation you are creating over here. Now you think the dung is going there. The dung has methanogens. They see anaerobic situation and they're like, yay, at last we have a lack of oxygen. We can do our action. So basically we are going to break down organic compounds and we are going to release something that we all need. And that is what? CH4. We are going to give down methane. Okay. We are going to give down methane, and then down this particular methane which is there, it has been along with methane there will be carbon dioxide, lot of things which are going to be released over here. In a gas holder, there is a gas holder also, okay, because these are volatile compounds, they will be going around, right? Now these guys are taken up by a gas pipe and it is provided to basically house or anything. Okay, you want to use it in household activity, you can do it. Okay, apart from that, you want to generate electricity out of it, you can do it. Whatever you want, you can do it. Okay, now ma'am, what will you do with the rest of the thing that is there? Basically this compound which is being made, now this still has a lot of important nutrients and stuff like that, because it is plant based thing. The cows are eating the plant. Okay, we have given the fruit covering and things like that, so it has lot of important compound. So it can be still used like manure. It can be still utilized like manure in the case of plants and stuff like that. So over here, what do you do? With the help of a pump or something. Okay, with the help of a pump or something, if it's in this particular region is in higher level, with the help of pump, you pump these things into the next chamber, and from here things can be utilized for formation of basically for making of the manure and things like that. Suppose this guy is over here and this guy is over here. So then with the help of gravity only it will be moving down like this. Okay, so this thing is kind of regular. I will be explaining you listen to me once more. Listen to me very carefully. So over here I have the dung plus the water. This is the mixing tank where I am going to mix it to form a slurry sort of thing. Now with the help of an inlet pipe, I am going to send it over here. This area is totally anaerobic. That means there is a lack of oxygen over here. Now over here, when there is lack of oxygen, these methanogens which comes along with the dung, they are kind of very happy. They are going to convert the organic matter into gaseous substances like CH4 and things like that. Now these gases are being transported to the gas holder, and then later through the gas pipe, it is sent to household areas or basically to produce electricity. Now you still have the sludge, primary sludge being present over here. What do you do with that? Basically this sludge can be used for acting as a manure, as things like that. So this can be taken up into the next area. Okay, and from here it can be used. So how do you pump it? So how do you take it down? With the help of a pump, or basically if it is in lower level, with the help of gravity only it moves down. This is what you mean by a biogas plant. This is how you have to make it. Okay, this is how you have to make it. If you think, okay ma'am, how do I make it? So basically let me help you with that. So over here we need an inlet pipe. Let's make an inlet pipe. It goes and acts over here. Okay, now over here we have a gas tank. This gas tank should be attached to a gas pipe. That is done, and this gas tank can go like this. Okay, now in the same area you will be having another box like thing which would be having the sludge. So your sludge is over here until here. So with the help of pump you can move it. So if you remember the mechanism of its working, you will remember the diagram also. As simple as that. This came as a 5-marker question, just imagine. Okay. Now how can you answer it? The biogas plant consists of a concrete tank, okay, which is 10 to 15 feet deep. Concrete tank is there, remember, concrete tank is there, it is 10 to 15 feet deep, in which bio-waste are collected and slurry of the dung is fed, okay, it is being put. A floating cover is placed over the slurry which keeps on rising as the gas is produced in the tank due to microbial activities. So floating covering is also done. Now the biogas plant has an outlet which is connected to a pipe to supply biogas to nearby houses. You saw the pipe. Used slurry is removed through another outlet and may be used as a fertilizer or manure. The biogas thus produced is used for cooking and lighting. The technology of biogas production was developed in India by the efforts of the Indian Agriculture Research Unit, okay, institute, not the unit. It is IARI, Indian Agriculture Research Institute, okay. Now, and Khadi and Village Industries Commission, KVIC as well. Now this is given as a part of the answer, so you have to follow it, the play. But anyways, if you're not even writing this, you're writing all this thing, that is also fine, okay. About floating cover also, if you're not writing, it's fine, because floating cover is never mentioned over here, so it's fine. Just explain them. There's a mixing tank, there's an inlet pipe, and this pipe that you see over here, what do you call this? This is an outlet pipe. Draw the diagram and explain in point by point. That is, just write first point over here, okay, make 2 over here, make 3 over here, and explain points. At first means this, second means it is going towards the digester. In the digester, this digestion is happening where the methanogens basically are getting activated and they are going to release methane, which is going to collected in the gas holder. From there it goes to the pipe to the household or for production of biogas, or basically it is going to act like what? As a source of electricity. And then you can say the remaining sludge which is there, it can be passed on to the next compartment, from where it can be used as a fertilizer or a manure or things like that. Easy. So write in points like this, it becomes all the more easy for you to understand and write as well. You don't miss any point as well. Cool. Chalo. Tell me the name of the organism that produces acetic acid. Okay. And we go on to the last question of the day, that is, explain the stages of sewage treatment. Now that is a fun thing to study, which is a big topic. Now listen, there is primary treatment and there is secondary treatment. What happens in primary treatment? Primary treatment means, for example, there is sewage. Sewage is what? The collection of waste materials and everything that you see on the roads and stuff like that. You take it and you are going to not put it in the river areas, because you know it will damage it. So basically you have to treat it, and then after treatment basically you can put down the water back into the river or any water source. So primarily the thing that you can do is probably separate the content manually, which you can see. So there is plastic and things like that, separate it. So there are 2 very important procedures happening up over here, okay, which is known as filtration, and the other one is sedimentation. Filtration basically means you have so many content, you have so many layers for things to get filtered. From here a content is being passed down. Okay, so what happens? All the small things which are there, it gets trapped. It will be getting trapped over here. Okay, whatever content is there, it gets trapped over here. That is what you mean by filtration. Okay, there are so many levels of filtration that is being taken, and then you have the normal watery thing is there. Now what is sedimentation? Sedimentation is basically according to its weight. Okay, for example, there is high weighted things, for example, there is stone in it and everything like that. They will slowly sediment down. So this gives you some fluids, this also gives you some fluids over there, okay, floating around over here. You take this thing and now you're going to treat it in the secondary treatment. What do you call this thing that you're going to treat? You call this as primary effluent. This is the sediment, the sludge that is being formed, you do not want it, okay. Basically you take this is known as the primary effluent. You got it from filtration and also sedimentation, you got this thing. The primary effluent is being now treated in aerobic condition with the help of aerobic bacteria. With the help of aerobic bacteria. Now, if aerobic bacteria is there, that means the BOD is high. The BOD is high, and during this phase only, I told you there are flocks being made. Flocks means what? Basically, the bacteria, the fungal filaments, and everything comes together. They make clumps, and it is known as flocks, which floats around, which are also going to break down a lot of things over here. So, BOD is kind of really high. So, basically, we put the organic matter. Okay, we have the aerobic bacteria. They will act on it. They will keep on acting on it. Okay, until and unless the level of BOD, it gets reduced. The level of BOD, it has to get reduced. Until that point, it is being treated. Okay, so primary treatment is basically involving filtration and sedimentation, where you're filtering stuffs and you are sedimenting substance. Whatever is left behind is known as primary effluent. Now you are going to treat it in aerobic conditions. Okay. In aerobic conditions, the aerobic bacteria is going to use a lot of oxygen, which is why the BOD is going to be really high. We will make sure that this particular process happens until the level of BOD gets reduced. And over here, bacterial and fungal filaments comes together, form clump. What do you call this? Flocks. Let's read it until now, and then we'll go ahead. Okay, so primary treatment involves the physical removal of particles large and small from the sewage through filtration and sedimentation. Initially, floating debris is removed by sequential filtration. So, with the help of sequentials, this sequence 1, 2, 3, with the help of that, you basically remove what? The floating debris, the floating things. Now, the grit, the soil and the small pebbles are removed by sedimentation. They will sediment down. All solids that settle from the primary sludge and the supernatant forms. Okay, so basically whatever solids they are going to settle down, they are going to form the primary sludge. So this thing that you go see over here, what do you call this thing as? You call this as primary sludge. And whatever thing is over here, it is known as primary effluent. That is what is said over here, है ना? Yes, and the supernatant. Supernatant means the thing that is left behind. It becomes the effluent. The effluents are from the primary settling tank. Okay, from the primary, this tank which was there, it was known as primary settling tank, taken for secondary treatment. So from the primary settling tank, where everything got settled, now we are going to take it for the secondary treatment. What happens there? The primary effluent is passed into a large aeration tank. So first was settling tank, where everything was happening over here. First was the aeration tank. Sorry, the settling tank where everything was settling down. Now you are putting down them to large aeration tank, where you are going to make sure that the effluent is going to have a mixture of air and everything. Now when it has a mixture of air and everything, the aerobic bacteria will come and help in growing. Okay, primary effluent is passed into large aeration tanks. This allows vigorous growth of useful aerobic microbes into flocks. The growth of microbes consumes the major part of the organic matter in the effluent. This significantly reduces the BOD, biochemical oxygen demand of the effluent. BOD refers to the amount of oxygen required to oxidize total organic matter. This we have studied. BOD is the measure of organic matter present in the water. Greater the BOD of wastewater, more is the polluting potential. Once the BOD of sewage water is reduced significantly, the effluent is now going to be passed for what? Passed into this settling tank, where the bacterial flocks are allowed to sediment. This sediment is known as activated sludge. Now again you are going to put it in next tank. You are going to put it in the next tank. First was settling tank, primary settling tank, then aeration tank, then now you are going to again put it in some other settling tank. And what are you going to put, my dear children? Basically, what did I do? Once the BOD is reduced, what I am going to do is that I am going to take this bacterial flocks and everything, okay, and I am going to let it sediment. I am going to let it sediment in the settling tank. Now this thing that is being formed is known as activated sludge. This is what, children? Activated sludge. So flocks and everything that I had, which was breaking down a lot of things, okay, whatever content was there, I was passing it to settling tank, all the things settled down. I call this as activated sludge. Small part of the activated sludge is pumped back to the aeration tank. A small part of it is pumped back to the aeration tank. Why? Because this has the bacteria and everything which will break down their things, no? So small part is just like how you're putting little amount of curd in the milk. Similarly, for more amount of curdling, similarly over here, you're going to put it there so that the process happens there also. This is acting as an inoculant. The rest of the things, the remaining sludge is pumped into anaerobic sludge digester. what are you going to do? You are going to put the remaining activated sludge into the next part, which is anaerobic sludge digester. Now it is anaerobic, my dear children, that means oxygen is not there. Now these anaerobic sludge digester, no, here you will help in the growth of anaerobic bacteria. There will be anaerobic bacteria, they will grow. Now they were just waiting for the situation to be like this. They will grow and now they will be feeding on the aerobic bacteria and everything which is present over here. We are going to feed on that. Okay, they are going to feed on that, and apart from that, basically they are going to release certain gases, which is what? The biogas. They are going to release what? The biogas as well. So the remaining sludge is pumped into anaerobic sludge digester. The anaerobic sludge digester, there is kind of bacteria which grows anaerobically, digests the bacteria and fungi in the sludge. During this digestion, bacteria produces biogas, which is a mixture of methane, hydrogen sulfide and carbon dioxide. The effluent from the secondary treatment plant is released into natural water body. So in the secondary treatment, no, okay, in the secondary treatment, when we were doing, we had, okay, when we were doing, basically anaerobic condition, everything was made. Out of this, the flocks and everything were taken to the next thing. But the effluent which was there, the water thingy which was there, it is being released to the water bodies itself, because this is safe now. This is very safe now. The remaining things which were there, you are calling it activated sludge, which is further treated. This effluent which is there, you're taking it and you're releasing in the water body. The activated sludge which is there, you are going to use this activated sludge, okay, for the production of what? Biogas. How do you do that? Basically, you treat it in anaerobic sludge digester. Anaerobic sludge digester. There, this particular guy, we have taken out the effluent from here, and now we are going to treat it and we are going to make gases which can be used. So I'll just repeat once, listen to me very carefully, okay? Can I remove this? I hope I can remove. Or should I make one? Okay, I'll make one. So first is primary treatment. Where what I am doing, my dear children, in primary treatment, I am going to physically remove all the content and everything. In that, I require filtration and I require sedimentation. At the end of it, you get something like this, where this is the pebbles and things like that. This forms the primary sludge and this forms the primary effluent. And this is happening in what? In settling tanks, in a large settling tank. Okay, this is the first thing that is being done. Now from here, this primary effluent is taken for secondary treatment. For secondary treatment, my dear children. Okay, over here what happens now? This particular whatever content is there, it is being put over here, which is why a lot of flocks and everything is being made. Okay, and this is aerobic condition. This is aerobic condition, and this is an aeration tank. This is an aeration tank where I am making sure, okay, where I am making sure basically I am producing enough amount of oxygen so that these bacteria and everything can grow together. What is this thing? This is the flocks being made, and the flocks are basically going to act on organic matter and break it down. Here the level of BOD was high, you have to make sure it goes low. That means all the organic matter is being broken down. Now what happens? This guy, if I pass it into the next thing, that is a settling tank again, I make sure that all this thing is being settled down, the flock and everything is being settled down, and the remaining content which is there, the watery thing which is there, it goes up. So this is an effluent. This is the next effluent that we are getting, and we are going to release it in water bodies. because this is safe. We have removed the organic content with the help of the flocks, but the flocks are still there. Now what we do, this particular thing, this structure which is being formed, what do you call this as? Activated sludge. Now to treat these flocks, I have to treat the activated sludge inside anaerobic sludge digester. I am going to make sure that there is lack of oxygen, there is no entry of oxygen over here. And now this activated sludge which is there, it is being treated with the help of anaerobic bacteria, which is going to feed on these bacteria as well as any waste which is there, which is still there if it is there, and it is going to give me gas, and this gas is taken up with the help of a biogas plant, a pipe, and that's how everything works. So you've given out the water, remaining thing you're using for what? Biogas formation. This is sewage treatment. If you want to take a picture of it, you can take, I'll just move aside. Okay, now I loved explaining this to you. If you think that basically you understood well, then let me know. I really want to know. Okay, so that's all, my dear children. With this is how you have to write the answer. Okay, write it properly, try to understand things, then only go ahead. If you're not understanding, you won't be able to write it as well. And with this, we are completing all the questions from this particular chapter. I think the numbering of the questions were kind of wrong. I'm so sorry for that. It was wrong. If it was wrong, okay? If it was wrong, I'm kind of really sorry for that. But anyways, so these are the things done. And if you are someone who is interested to join our test series, which would include more questions, more practice and everything, then I cordially invite you to this. You can join our test series by paying 599 rupees. The link is given in the description box. Go through the content really nicely and then only go ahead. So over here we provide 12 mock tests, my dear children, where the 4th of them will be part test. Initial 4 of them will be part test. That means in a test paper you will find multiple subjects, okay, combined together, and then there will be 8 full length tests. That means individual tests will be there for chemistry, for bio, for things like that, okay. And people who have opted for Kannada, they have to pay a little extra, otherwise this is the normal number, and you get video solutions for that. The first test series will be launched tomorrow on 15th of December, so be active there. So that's all, my dear children, and if you think this video was really cool, good, definitely do not forget to tell me, and do not forget to subscribe to our channel, and I wish you all the very best and all the love in the world. Bye bye, take care.